Hi.
Hello. Okay, before we get started, are there any questions?
No questions.
Okay, today is May 31st, and we're supposed to be working on chapter 13, viruses, viroids, and prions. We will talk about that topic all day today, and hopefully we'll finish it on Thursday. There is no lab this week, but I will be logging in from 6:30 to 6:45 to answer questions. So if you have a question, come between 6:30 and 6:45 and I'll try and answer those questions. You should be working on your unknown work. Some of you have finished. Some of you haven't gotten started. I have to say, you're really late, and I doubt you'll be able to finish the project. But some of you are close to finishing, and so work on your unknown work. Okay? Any questions about any of that? The unknown project is due Saturday, June 11th. Your unknown notes pages will close Friday, June 10th. So the last day I will take a test will be Friday, meaning a test request. And if you give me a test request on Friday, it has to be a test that can be done within a day. And there's only a few, like the catalase test is one. If you give me a 2-day test, I'll tell you, sorry, there isn't enough time to incubate it. So really, the last day you should be asking for most of the tests is Thursday. Any questions about any of that? And let me remind you, there are some tests that take longer than 2 days. The MR test takes 5 days to incubate. All right, there's no questions. I'm going to move into the lesson. We talked about the viral structure, saying that viruses have a genome that's made of nucleic acid, but that genome can be either DNA or RNA. And the DNA or the RNA can be double-stranded or single-stranded. Another part of the viral structure is all viruses have a capsid, that's the protein coat, surrounding the viral genome. So the viral genome's the innermost molecule, and then there's a protein coat surrounding the viral genome. The capsid is made of protein. Blow this up just a little bit more. Over. And the protein, each one we call a capsomere. So that would be a capsomere, and that would be a capsomere, and this would be a capsomere. And this viral coat is only made up of one protein, or one capsomere. But that doesn't have to be the case. You can have a virus made up of the capsid made out of several different proteins. And if the virus has a head and a tail, the head is usually made of a different protein than the tail. And we'll take a look at some of those viruses in just a little bit. This virus has several, whoops, went too far. This virus has several proteins in it, and it also has something more than more than the capsid. But with that capsid there, if we blow that up, let me see if I can blow that up a little bit. Up too far again. So there's the genome here, and then there's the protein coat, the capsomere. And this one has a viral envelope around the protein coat. Viruses may have an envelope, but not all viruses have an envelope. And the envelope is on the outside of the protein coat that we call the capsid. Any questions about any of that? If the virus has an envelope, it may have spikes. That would be these pinkish purple lumps here. And the spikes cover the envelope. The spikes are usually glycoprotein, although they could be proteins. And they are antigens that the host recognizes on the virus. Now, an antigen is just a molecule that the host uses to recognize that the molecule or the virus or the infective agent, the host recognizes as foreign, and it does that by certain molecules that we call antigens. Antigens are the outermost molecules of an object. And well, the host uses those molecules to recognize the body as being foreign to the host. And those molecules we call antigens. Any question about any of that? So an antigen is just a molecule that the host immune system recognizes and recognizes as foreign. And then it'll mount an attack, an immune response against that agent. So antigens for a virus are always the outermost molecule. And if the virus has spikes, the spikes will be the antigens. But on this virus that doesn't have spikes because it doesn't have an envelope, what is going to be the antigens that the host will recognize as molecules not belonging to the host? Where will the antigens be? All right, we'll think about that. We'll come back to it. So when we're talking about viral structure, it's important to realize that viruses have specific general morphology. That means there are three basic shapes, and we classify viruses on the shape of their capsid structure. There are helical viruses where the capsid is in a helix. And I personally don't see the helix in this one, but I can see it here in this helical virus. There are polyhedral viruses, and these are viruses with many sides, sort of like a complex dice. If you've ever worked on a dice that's more than the normal six-sided dice, I'll word it that way, that's polyhedral. And there are viruses with that type of a shape. Here's one. This is actually a envelope virus that has spikes on it, but still the capsid is in a polyhedron. And then there are viruses that we call complex viruses. They can take on essentially any shape. Like this is a complex virus, and there's a complex virus. And there's a complex virus. If you don't recognize that, that sort of looks like the Apollo lunar lander. And that's actually a picture of a real electron microscopic image of this virus here. So a complex virus can take on essentially any shape. Any question about that? One complex virus that we'll talk a little bit about is bacteriophage T4. I think I mentioned that a bacteriophage is a virus that infects a bacteria. Have I mentioned that? All right, nobody's responding. I don't know what that means. Anyways, bacteriophage T4 is the most studied of all viruses. And the reason why it's the most studied virus is simply that this virus was one that infected E. coli, and E. coli was something that microbiologists had easily available to them in the lab. So they frequently studied bacteriophage T4 simply because it grew in E. coli. Any question about any of that? Excuse me a minute, I need to blow my nose. Hopefully you didn't get to see any of that.
Excuse me, I have a question.
Uh-huh.
So would the COVID-19 virus, would that be the envelope virus?
COVID-19 is an envelope virus, yes.
Okay.
Okay. Yeah, we'll talk a little bit more about COVID-19. And you know, I should have updated my webpage, but I'll probably talk about COVID-19 tomorrow, not today. So I still have a chance to update my webpage. The trouble with COVID-19 is I'm talking a little bit about it, just 3 or 4 slides. And we're still learning about COVID-19. So the information is constantly being updated and changed. In particular, it's the lethality of this virus changes. The different variants have different lethality. All right, so T4, oops, boy, I got that really small. T4 is a complex virus, and it belongs to the T even family, and they all have this basic shape, and that's T2, T4, and T6. They're all bacteriophages. I did mention a tail. This part of the virus is a tail, and it's composed of different proteins than the head. There's probably only one protein in this head, part of the capsid. This is part of the capsid, but we call it a tail. And this probably has at least, I would say, 3 or 4 different proteins. For the structure here, there's one, and then there's one for the tail fibers, one for the plate, and then one for the sheath. So this tail has several different proteins. What do I want to say about that? Yeah. Shrink that just a little. So infection of a virus with an animal causes the animal or the host to respond to the virus. And the host immune system will usually recognize the virus as foreign and then mount an immune response against it. And when a virus infects an animal, the animal will usually produce antibodies in response to the virus. And then the antibodies will attack the virus. Antibodies recognize and bind to the viral surface proteins. And these molecules I've already mentioned we call antigens. It's what the host recognizes as foreign, and the antibodies will bind to the antigens. One problem that the host has in fighting the virus is that some viral proteins can change or mutate frequently. And then there's different viral strains, and therefore they can infect the same individual more than once. As you know, an example of this is COVID-19. And there have been several reported cases of people who have been infected at least 3 times, even sometimes 4 times, by COVID-19. And what's happening is the viral proteins are changing the antigens, and therefore the virus can infect the same host repeatedly. If the virus were not to change, meaning the antigens don't change, then when the host immune response responds to the virus, the patient would be immune, and then the patient would never get another infection. An example of that would be polio. I don't know if they even still give the polio vaccine, but I got the polio vaccine. And that polio vaccine was made in the 1950s, and it's still being used today. So at least with the polio virus and many viruses, they don't change frequently. And then the vaccine that we develop is good for a long time. The 1950s was that 70 years ago? I believe it was. And that vaccine is still good for the polio vaccine. And fortunately, it's true for many viruses like the mumps and the measles, where the vaccines are still good. But as you know, COVID-19 and the flu change, and the vaccine is only good for at best a year, maybe only 6 months. And we'll talk a little bit more about that when we're talking about COVID-19. And we'll also talk a little bit about it with the flu, when we talk about the flu. Any question about that? That is a reason why you should get a flu shot about every year. You don't have to get the flu shot every year, but you do have to get it at least every 3 years, because the flu changes enough that after 3 years, the flu vaccine will no longer be good. Any question about any of that? All right. So when we're talking about viruses, we do put them into a taxonomy, different groupings, and viruses are grouped into families. There is, let me see if I remember that right. Is it in order? Peter's son came over from, yeah, order. There are some viruses who have an order, but most viruses we're just going to call or grouped into families. They're based on their type of nucleic acid, the replication strategy the virus has, the morphology of the virus. And the higher groupings, other than the kingdom that the virus infects, we don't group viruses into them, those groups. And the reason is we don't know the taxonomy of the virus. And the reason is that the viral genome is small, and it changes rapidly too, or fairly rapidly. And so we don't know the evolutionary history of viruses beyond the family. And sometimes we do know the order, but beyond the order, we don't have the viruses grouped other than the kingdom that they infect. So all viruses have a family, and the family ends in the name Viridae. All viruses we break from family into a genus, and the genus names always ends in virus. We then have a viral species, and this will be the name of the virus, a group of viruses sharing the same genetic information and ecological niche, meaning they infect the same host or hosts. And we use common English names to name the viral species, such as COVID-19 or the human herpes virus. That's actually a genus, but you could say human herpes virus simplex for the viral species. And then we have subspecies, and they're designated by a number. For humans, we could call the races of human a subspecies. Okay. So like a European would be a subspecies. And with viruses like human herpes virus type 1, that would be a subspecies of the herpes virus, the species, and then of the genus herpes virus. And then the family would be family herpes viridae. Okay. So you'll be pleased to know when we're naming viruses that common English names are used. We do not have Latin names for viruses. And it's just that the early virologists were all English speaking, and then they started naming the viruses. And then the other languages, like the British and the German virologists, came along and they simply used the same common English names that we have. So hopefully you will like that. So we have the polio virus, that's a common English name, and that would be the virus species. Now let me tell you how important it is to have a subspecies. With the polio virus, that's the viral species, it can cause the disease polio. And that would be one of the subspecies of the polio virus. But there's another subspecies of the polio virus that we use as the live vaccine against polio. And it is simply a subspecies of the polio virus. It's the, let's see, not the Salk, Sabin. I think it's the Sabin vaccine for polio. All it is a different subspecies. And it doesn't cause the disease polio. We use it as a vaccine to prevent the disease of polio. So that tells you how important the subspecies are in regard to the diseases they cause. But they're all the same viral species. And in fact, polio 1, meaning the one that causes polio, and polio 2, the one that we use as the oral vaccine to prevent getting polio, they all only live in humans. And so they only reproduce in human cells. And so they do share the same ecological niche. It's just one does not cause the disease polio and the other one does. All right, any questions about any of that? So the family would be herpes viridae, the genus simplex virus or human herpes simplex virus, more likely what we call it. The common name, human herpes virus. This is a scientific name that most people have not been using, and it doesn't look like it's going to stick. Most people are calling it the common name, herpes simplex virus. I'm sure you've heard of that. And then the subspecies would be human herpes virus 1 or herpes simplex virus 1. And this would be the virus that causes cold sores. Any question about any of that? All right. Let's go back just for a minute and say another subspecies of this would be herpes simplex 2, which causes genital lesions. So it's the same species, just another subspecies. One causing cold sores and the other causing genital lesions. All right, for growing bacteriophages, they have to be grown inside of a cell. All viruses can only grow inside of their host cell. And for a bacteriophage, it has to grow in a bacteria cell. Bacteriophages are usually grown in bacteria. And what we do is we will plate the plate with the bacteria. And that's why the plate looks whitish right here, because there's bacteria making it whitish. Right there and there, these are what the bacteria looks like once it's been lysed. And what we do is we put on a solution on this plate containing bacteriophages. And if the phage attaches to the bacteria, it'll then replicate. And if the virus replicates and lyses the bacteria cell, it'll then go out and attach to other bacteria and then infect those cells, lyse them, and then infect other cells. And eventually you'll see plaques. This is a region where all of the bacteria in this area have been lysed from the bacteriophage. Any question about that? I've actually grown bacteriophages in a plate like this, where you count the plaques, and I don't know how many this is, maybe 200. So the solution of bacteriophages in this solution we put on here would about been 200 plaques per mil, assuming you put 1 mil on the plate.
So to grow the bacteriophages, do you plant like the cells already with the bacteriophages, or do you put the cells on the plate and then you put the bacteriophages on?
You first put the bacteria on the plate, but don't allow the bacteria to grow yet. And then you put the solution with the bacteriophages on it so that the phage can attach to the bacteria. And then you grow the two together. And where there's no viruses, you'll get the plate looking white like that. And where there's a virus, you'll get the plaques. Okay.
That's interesting.
Yes.
How do you isolate the virus from living cells, though?
Well, you can centrifuge the bacteria, and they will go to the bottom of the centrifuge tube, and the viruses are much smaller, and so they would be in the solution.
Okay.
Okay.
That's
So
interesting.
that would be one easy way to separate bacteria and viruses.
That's interesting. Thank you.
All right. I'll shrink that down a little bit. So let's talk a little bit about viral multiplication. The virus, the genome only contains a few genes, meaning viruses don't have much genetic information. And these genes will be the genes that the viruses needs to replicate. They will be the genes for the coding for the viral proteins and maybe a few viral enzymes. And the purpose of the virus will be to hijack a cell and then use that cell to replicate the virus. So following infection, the virus takes over the host cell's metabolic machinery and then uses the host cell's ribosomes, tRNA, amino acids, ATP, and some of the host cell's enzymes to replicate the virus. The basic mechanism for viral multiplication is similar in all viruses. For bacteriophages, remember this is a virus that infects a bacteria cell, and it's sometimes shortened to phages or phages. I won't use that term. I'll always use bacteriophages. These are viruses that infect bacteria cells. They have only two mechanisms of reproducing, and that's the lytic life cycle and the lysogenic life cycle. In the lytic life cycle, the virus replicates in the host bacteria cells and then lyses the host cell. Obviously, it kills it and then lyses the cell. And when the cell is lysed, that releases the newly replicated viruses out of the cell so that they can go on and infect other bacteria cells. In the lysogenic life cycle, the virus can replicate, but when it's replicating, at least in the lysogenic life cycle, the host cell remains alive. So the virus and the host cell are living in symbiosis. And yes, that bacteriophage can replicate in the lysogenic life cycle. Any questions about any of that? So before we talk about viral multiplication, I want to ask you, what does a bacteriophage, what do we need? What components does the virus need to make in order to replicate?
RNA, DNA?
Yeah, it needs to make its replicate its viral genome. And that could be DNA or RNA, it doesn't matter. It does need to replicate its genome. What else does the virus need? All viruses need at least two things to be replicated, one being the genome. All right, nobody's answering, so I'll answer it. You need to replicate the viral protein code. So you need to take the viral DNA and then replicate it to make new viral DNA. And then you need to transcribe that viral DNA to make viral messenger RNA, and then translate that viral messenger RNA to make the viral protein, specifically the capsid proteins, and then get the capsid protein together with the viral genome to make the new progeny virions. So at the very minimum, that's what we need to replicate in a virus, the viral genome and then the viral capsid proteins. In the lytic life cycle, there are 5 stages for the virus to replicate. And I know that won't work. Well, maybe this will work because this is from my web page. Let's see. I think I have to. I don't have this. Just a minute here. I got to put my thumb drive in here because I don't have this movie. I don't have the movie on my computer, so, but it's on my thumb drive.
First, the phage particles stick to the surface of the bacterial cell. They bind to specific sites called phage receptors.
Well, that isn't working, so let me try the other movie and see if that one works. Hopefully you'll be able to watch that from your own computer, but if not, well, that's not good. We'll be able to hear it.
In a way, a virus is a parasite. It attacks a host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle.
Okay, any questions about the movie? Let me leave my thumb drive in here because I've got another movie, or the rest of this movie we'll talk about in a little bit. Can you guys hear me okay? I'm not using the headphone because I can't have the headphone and my thumb drive plugged into the computer.
Yes.
Okay, good. All right. So in the lytic life cycle, the first thing we have is attachment. This is when the bacteriophage attaches to the host cell, which in this case will be a bacteria cell. The next stage is penetration, where the phage lysozyme opens up the cell wall. You need to make a hole in the cell wall for the virus to infect the cell. Yeah, let me go to the movie on that just to show a little bit about that. See if I can find it. That looks like I can't.
And the nucleic acid from this capsule is injected through the hollow. Certain type of bacteria can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall.
So it uses the enzyme lysozyme to digest a little hole in the cell wall.
And the nucleic acid from this capsule is injected.
All right, and so to infect the nucleic acid from the virus, the sheath actually contracts, and then that puts pressure on the contents in the head, and so that injects the nucleic acid from the virus into the cell. Any questions about any of that?
Yes. Is this only for the bacteriophage, or is it for every virus?
It's for a lytic virus, but and it's not for all. Let me see, think about that. You know, it's for most, if not all, bacteriophages, that they do inject the genome into the bacteria. For animal viruses, it's much easier to infect an animal cell, and we'll talk a little bit more about that. So animal viruses do not inject their genome into the host cell. So I guess it's mostly for bacteriophages.
Thank you.
Sure. And that tail sheath actually contracts, and then that forces the contents of the head into the cell, much like a syringe works. You contract the needle, and that pushes the contents of the syringe into the patient. And then the virus goes through biosynthesis, the production of new bacteriophage DNA and bacteriophage proteins. And then you have maturation, the assembly of the new virions, meaning the protein coat gets together with the viral genome. And then you have release of the virus. And that was really done well in this video here. Let me see if I can find that.
organelles to replicate new viral virions. Finally, the muphages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions.
Sorry about that. I don't even know how to get rid of that. There it goes. The enzyme that's released by the virus to weaken the cell wall is once again the enzyme lysozyme, and that is an enzyme which digests peptidoglycan of the bacteriophage cell wall. All right, let's go to where we were because we'll.
Is called the lytic cycle.
There we go. I'm going to shut that one down, but how do I get it? There we go. Yeah, you'll have a hard time understanding that, but I'll let you view that on your own. So there we saw the lytic life cycle. After the lysozyme breaks down the cell wall, the cell bursts, and that releases the newly replicated viruses. I think I've talked about all of this, the lytic life cycle. So let me just make sure. Yeah. All right. So 2 terms you should know for lytic life cycle viruses. There's the burst time. That's the time from when the host cell is first infected to the time where the host cell bursts, called the burst time. And that'll vary with the species and with the temperature. But for most bacteriophages, the burst time is between 20 and 40 minutes. And then there's the burst size. That's the number of newly released viruses from the cell, meaning the newly replicated virions. And you don't need to know the number. It's usually between 50 and 200. It'll depend on the species of the virus. All right, any questions about the lytic life cycle, where the virus replicates in a host cell, and when it's done in replication, it lyses the cell, why it's called the lytic life cycle? If not, let's talk about the next one, and that's the lysogenic life cycle. Yeah, with a bacteriophage, which follows the lysogenic life cycle, it will infect a bacteria and then inject that bacteria into the host cell, where one of 2 things has to happen immediately, and the virus decides what it's going to do. The first thing it can do is it can follow the lytic life cycle. So a lysogenic virus can follow the lytic life cycle, okay? Or it can decide to follow the lysogenic life cycle. Any question about that? So a lysogenic virus, it immediately has to decide once it infects a cell, will it follow the lytic life cycle or will it follow the lysogenic life cycle? All right, the lytic life cycle happens as we've previously mentioned with the lytic life cycle, where the virus hijacks the cell and uses the cell to replicate more copies of the virus, and then it'll lyse the cell, releasing the newly replicated viruses. And then that newly replicated virus will attach to another bacteria and then infect the cell. And then once again, it has to decide, will that virus enter the lytic life cycle or will it enter the lysogenic life cycle? If the bacteriophage decides to enter the lysogenic life cycle, then the phage will remain inactive, and we'll call it latent. And it'll enter the lysogenic life cycle, and it remains in the lysogenic life cycle for as long as the lysogenic life cycle is continuing. Okay? Any question about that?
Yes.
Let me go ahead and tell you that what determines which cycle it's going to go into is there's a repressor protein of the lysogenic life cycle. And if that repressor protein is active, it shuts down the lytic life cycle. It shuts down the lytic life cycle genes of the virus, and then the lytic life cycle does not continue. If that lysogenic repressor protein is not active, then the lytic life cycle follows by default. Okay, and then the virus never enters the lysogenic life cycle. So you have to have that gene turned on, and then it'll switch off the lytic life cycle, and then we enter the lysogenic life cycle. All right, let's watch a little video on the lysogenic life cycle. And it was actually this video here. So this is both the lytic life cycle and the lysogenic life cycle. And we only have seen the lytic life cycle. So let's watch the lysogenic life cycle now.
Interestingly, the lytic cycle does not always happen immediately. Sometimes, rather than producing virions, phage nucleic acid incorporates in the host cell DNA. Now, each time the cell divides, the phage DNA is also reproduced and is spread to more and more cells. Then suddenly, for reasons that are still unclear, the phage DNA activates and the destructive lytic cycle begins. Once again, the host cell is destroyed, and a new generation of phage are released to invade new hosts. In a way,
Oops, sorry. All right. I really like this movie because everything about it is correct, and it's very short, and it mentions both the lytic life cycle and the lysogenic life cycle. There's only one thing that's slightly wrong, and that is, see this virus here?
a virus is a parasite.
It looks like virus T4. And virus T4 only follows the lytic life cycle. And this one is following both the lytic life cycle and the lysogenic life cycle. And to be a lysogenic virus, you cannot be T4. But other than that, that's the only error in this movie, so I've kept it. And the sound is a little bit off, but it's such a good movie, and I can keep this on my own computer and put it on my web page so you guys can watch it. So I've kept it, okay? And I simply downloaded it many, many years ago from the internet. All right, how do I get rid of this? There we go. So let's talk a little bit more about the lysogenic life cycle. So the virus has infected the cell, injected its nucleic acid into the cell. And the first thing a lysogenic virus has to do is decide, do I want to enter the lytic life cycle or do I enter the lysogenic life cycle? If it enters the lysogenic life cycle, there is a repressor protein that keeps the lytic life cycle dormant. And then the lysogenic genome, let's make it a DNA virus. The DNA virus will incorporate into the host cell's chromosome. So this dark purple is the bacteria chromosome DNA, and then the pinkish purple is the viral DNA. The two come together. and make one molecule. We call the viral genome in the host cell's chromosome, we call that a prophage. So only a lysogenic virus can have a prophage. That's the viral DNA incorporated into the host cell's DNA. Now, this virus can replicate in the lysogenic life cycle. And how it replicates is whenever the cell divides, it will replicate the chromosome, and that will replicate the viral genome. So instead of having one virus genome here, we now have two. And that cell will go through division. And so we'll have two cells, each with the lysogenic viral genome. And that can go through many cell divisions this way. Okay. Now, as the video told you, occasionally for reasons that are not fully understood, even after 70 years of studying lysogenic viruses, they still don't have a good handle on this. But occasionally, the lysogenic virus will leave the lysogenic life cycle and enter the lytic life cycle. If that happens, the prophage always excises itself from the host chromosome. And then the lytic life cycle will begin. So from this cell, we then will enter the lytic life cycle, where the viral genome will be replicated, where we have several copies made of it. And then we replicate the lytic capsid proteins. And then we assemble the viruses, you lyse the cell, and then that releases the newly replicated virions. We still call this a lysogenic virus, even though it lysed the cell. And when it infects the host cell, it then has to decide, does it want to enter the lytic life cycle or enter the lysogenic life cycle? Any question about any of that? All right, so I mentioned that we don't really understand most of the time why the virus leaves the lysogenic life cycle and enters the lytic life cycle. But they have discovered one thing that does make a lysogenic virus enter the lytic life cycle. And that is if you damage the host chromosome, like subject it to a mutagen like X-rays, then this virus will realize that the host chromosome is damaged and that the host cell may die. And so the virus will leave the lysogenic life cycle and enter the lytic life cycle. But other than that, we don't know why a virus leaves the lysogenic life cycle and enters the lytic life cycle. And they've been studying this for over 70 years, and they still don't have a good understanding about that. Any questions about that? All right, if there's no questions about that, let me mention that there are some consequences of lysogeny. First, a bacteria cell that has a lysogenic virus living in it is immune to infection by the same bacteriophage, meaning one bacteriophage infects this cell, other bacteriophages cannot infect this cell, at least of the same species. So that cell is immune to further infection by that lysogenic virus. And then the second consequence of lysogeny is phage conversion can occur. This host bacteria cell gets new DNA, and that's DNA of the virus. And this new DNA can give this host bacteria cell new properties because it has new DNA. A specific example of this is Corynebacterium diphtheria only makes the diphtheria toxin when the bacteria is infected by a lysogenic virus. And the reason is the Corynebacterium diphtheria toxin is encoded by the genome of the lysogenic virus. And so any Corynebacterium diphtherium that does not have the lysogenic virus cannot make the diphtheria toxin. It only makes the diphtheria toxin when the bacteria has a lysogenic virus. So yes, depending if the bacteria that's infecting you is infected with the virus, can determine how sick you will become. Any question about any of that? All right. So the thing is that the bacteria cell can get new properties from the genes of the lysogenic virus. All right, enough about bacteriophages. Let's talk about animal viruses. How do they replicate? And you know, I think if I shut down this movie, which I've already done, let me go ahead and shut that down too. I can take out the thumb drive and put my headphone back in. So animal viruses are viruses that infect animal cells. And I did mention that we do classify viruses into the kingdom that they infect. There are bacteria viruses that we call bacteriophages. There are animal viruses that infect animal cells. And there are plant viruses that infect plant cells, etc. Animal viruses follow multiplication of the virus similar to the bacteriophage viruses. There are some differences, but the attachment still happens pretty similar, and that is the viruses attached to the cell membrane, and there are specific viral proteins that we call ligands or attachment molecules that attach to the host cell molecules, and those we call receptors. Animal viruses are a little different, and that is they attach to the cell membrane. Obviously, a bacteria virus has to attach to the cell wall of the bacteria. The penetration is different. We'll talk a little bit about that. It happens by endocytosis or fusion. The encoding is a little different, but it can use either viral or host enzymes. The encoding is how the animal cell gets the viral genome into the cell. The animal cells don't inject the genome into the cell. Biosynthesis is pretty much the same as bacteriophage biosynthesis. Maturation is once again similar. And then release can happen by the lytic life cycle, meaning there are animal viruses that follow the lytic life cycle. And that is one way the animal cell can release itself, but they can also release themselves from budding or something similar to budding. Okay, so there are different ways that animal viruses can release themselves. When we're talking about animal viral replication, the replication for animal viruses is similar to the replication for a bacteriophage. There are several differences. There's only one that you really need to know about, and that is for bacteriophages, they can only replicate in the bacteria cytoplasm, because in bacteria, that's all there is, cytoplasm. For animal viruses, they can replicate in the animal cell cytoplasm, but they can also replicate in the nucleus, in which case they're not replicating in the cytoplasm. They're replicating in the nucleoplasm. Any question about any of that? And I'll give you a general rule of thumb. Hopefully you can see my thumbs. And that is a DNA animal virus generally replicates in the nucleus, and then an RNA virus replicates in the cytoplasm. Why they do this is a DNA animal virus needs the machinery of the nucleus to replicate. So the DNA virus replicates in the nucleus. And then the RNA animal virus needs this machinery of the cytoplasm to replicate. So generally it replicates in the cytoplasm. There is only one exception to that rule of thumb, and that is retroviruses are an RNA virus, but they do replicate in the nucleus. And you'll understand why when we talk about retroviruses, which will not happen today. That'll happen on, is it Thursday? In our next lesson, okay? I'll talk about retroviruses in our next lesson. You should also realize that with the multiplication of animal viruses, when they attach to the cell membrane, they penetrate the cell membrane by either endocytosis or fusion. They do not inject the nucleic acid into the cell. They enter the cell by either endocytosis or fusion. Endocytosis is when the virus attaches to the host cell, and then the host cell invaginates around the virus we're going to talk about, and that will grow, and then this will form. The membrane will pinch off right here, and then the cell membrane will reform across here, and then this will form an organelle inside the cell and bring the virus inside the cell. That is endocytosis. The other way that viruses can enter the cell is by fusion. And let me actually draw it here because I don't have a good picture of it here. What happens in fusion is that the lipid membrane of the animal cell can fuse to the lipid membrane of the cytoplasm. I'm going to try and draw that. There we go, paint. This out. So here we have our virus. Not a very good picture. It's supposed to be round. We'll just make a huge cell. The two attach. I'm going to move this down. And this is going to be difficult for me. Let me get the pencil. They attach, and let's draw it over here, a blow up. The cell being like this will then fuse with the membrane of the virus, and then the double membrane of the cell actually combines with the membrane of the virus so that we have it looking like this, sort of. You get the picture? And then this can be brought inside the cell, and this is the virus right here. And that's by cell fusion, where the membrane of the virus fuses together with the cell membrane. Any question about that? I tried to draw it. I'm not a good artist, but hopefully you understood. So penetration can happen by endocytosis or fusion. Endocytosis, this invaginates, and then that's brought into the cell that pinches off. And then you have a release mechanism that we're not going to get into. It'll remove the envelope of the phagosome, and then the virus will be degraded, meaning the capsid protein will be degraded, and then that will release the viral genome into the cell. The other way is if this were to fuse to the cell membrane, and then the cell membrane would actually be part of the viral membrane, and then it would go around, and that would bring in the virus into the cell. We're not going to talk about encoding, but it can happen by either viral or host enzymes. And then biosynthesis would be the same in the bacteriophages. You have to replicate at a minimum the nucleic acid of the virus, and then the capsid proteins of the virus. Maturation will be similar to bacteriophages, where the viral capsid proteins get together with the viral nucleic acid to make the new virions. But release can be different. As we mentioned, we can have a lytic animal cell virus, which follows the lytic life cycle. An example of an animal virus that follows the lytic life cycle is the polio virus. Follows the lytic life cycle. Another one would be the human cold. Follows the lytic life cycle. But we don't have to have that for animal viruses. We also have the budding life cycle, where an animal virus replicates generally at low numbers in a host cell. And then the viral DNA replicates and gets together with the viral capsid. And then that new protovirion, because not finished, we'll call it an early virion of the virus, will actually come up to a membrane of the cell and then start pushing on it. Okay, and it actually comes up to a membrane that has viral proteins in it. So we see these blue things in the membrane here. In this case, it's the plasma membrane, but it doesn't have to be the plasma membrane. It can be the plasma membrane, it can be the nuclear membrane, or it can be the envelope of the endoplasmic reticulum. Okay. So it comes up to a membrane and then pushes on it, and it comes up to a membrane that has viral proteins in it. And then it pushes on it and pushes on it, and then it rips off a part of the membrane. That membrane generally reforms, so the cell doesn't die if that's the nucleoplasm or the cell membrane. It reforms, so the cell doesn't die. And then that releases the newly replicated virus. And this part of the membrane that's ripped off by the virus forms the envelope of the budding life cycle virus. And that is how envelope viruses obtain their envelope. And that is they follow the budding life cycle and they rip off a portion of the membrane of the host cell they've infected. And that part of the membrane becomes the envelope of the budding life cycle virus. Any question about any of that? All right. So all enveloped viruses follow the budding life cycle. And that envelope is either from the membrane of the cell membrane, the nuclear membrane, or the endoplasmic reticulum. And it depends on the virus which membrane it rips off to get the envelope. If this is the cell membrane, you can now see that this is released by the cell. And that's the release of the virus. This can now go on and infect another cell. And here we see a picture of a cell which is forming buds. And that's a budding life cycle virus. Any question about any of that? Oh gosh, I got that picture again. Sorry, if I come over too far to the right, that picture comes up. All right, viruses have different genomes. They can have a DNA virus or an RNA virus, and they can be double-stranded DNA virus, or single-stranded DNA virus, or double-stranded RNA virus, or single-stranded RNA virus. Each of these viruses needs to have a slightly different mechanism for multiplication and biosynthesis. Obviously, double-stranded DNA virus will replicate similar to double-stranded DNA of the host cell. So we've already talked about this one, and that is the double-stranded DNA simply separates, and then one strand is used as the template to replicate the other strand. Okay. And the same thing with transcription. We'll separate this, and then one strand will be transcribed to get the viral gene transcribed on that genome. Any question about that? All right, let's briefly talk about the DNA viruses. There are 7 families, 1, 2, 3, 4, 5, 6, 7 families of DNA viruses that infect human cells and cause disease. So there are 7 viral families of DNA viruses that cause human disease. You do not need to memorize these families. We'll talk about some of them, like the herpes viridae, herpes virus family we've already talked about. And we'll talk about the pox viruses next time. I'm not sure. We'll talk a little bit about adenoviruses and maybe the papilloma viruses a little bit. They each have DNA for their genome. This tells you what type of DNA it is, double strand or single strand. Interesting, this one is both partial single strand and partial double strand. Most of them are one or the other. And then it tells you if they're envelope virus or naked virus. Hopefully I mentioned last time that a virus, if it doesn't have an envelope, we call it a naked virus. And some other information about the virus, and then namely the different diseases they cause. The pox viruses cause smallpox. And we'll briefly talk about smallpox and cowpox a little bit too. Herpes viridae, the herpes virus diseases, we'll talk a little bit more about them. And that's about it. Any question about the DNA viruses? Don't memorize this table. I'm just showing you the different families that cause DNA viruses. They contain obviously DNA. They infect animal cells. That's why they're animal viruses. DNA, animal viruses, and actually, well, yeah, DNA animal viruses as well as DNA plant viruses are generally replicated in the nucleus, and it usually uses viral or host enzymes, and it uses the machinery of the nucleus to replicate the DNA virus. The capsids are synthesized in the cytoplasm, and then the capsid proteins are got together with the newly replicated DNA to replicate the virus. Okay, any question about any of that? All right, let's talk about when a DNA animal virus enters a host cell. It has to decide, am I going to enter the lytic life cycle? And if it is a lytic life cycle virus, that's the only life cycle it can enter. But if it's not a lytic life cycle virus, it can enter another life cycle, such as the budding life cycle. And if it does that, it has to enter the host cell chromosome. And if it does that, we call it not a prophage because it's not a bacteria virus. It's an animal virus that's entering the host cell's chromosome. And that we call a provirus. Okay. If the DNA of the virus ever enters the chromosome of the host cell it infects, that provirus will remain a part of the host cell DNA permanently for the lifetime of the cell. The provirus is never excised from the host chromosome. And the provirus can be replicated or will be replicated when the host cell replicates its DNA. Any question about any of that? All right. If the viral DNA remains outside the cell and then goes through the lytic life cycle, it will replicate its DNA, making new virion DNA. And I'm only showing it for double-stranded DNA, but it would be similar for single-stranded DNA. It'd be a little different for RNA, and I'll show that later. But the genome gets replicated. The genome will be transcribed into viral messenger RNA. The viral messenger RNA will be translated into the capsid proteins. And then the capsid protein has to get together with the newly replicated viral DNA to form the new progeny virions. And if the virus is following the lytic life cycle, it will lyse the cell to release these new progeny virions. Any question about any of that? All right, I think we will stop right here and I will continue with this next time. Any questions? All right, if there's no questions, I will see you next time. I will be in the lab from 6:30 to 6:45 to answer questions. So log in during that time. After 6:45, if there's no questions, I'll log off. All right, I'll see you.